Good morning. My name is R. Ritish Rathod from Department of CSC AI/ML Section C. Coming to my course title, Essential of Problem Solving, and my today's topic is Applications of Hamiltonian Cycle in Bioinformatics, a survey. Today I am going to discuss this topic, Applications of Hamiltonian Cycles in Bioinformatics, a survey. Coming to its introduction, cycles are fundamental concepts in graph theory involving a cycle that visits each vertex exactly once. Their applications in bioinformatics had gained attention due to the complex nature of biological data. This presentation surveys the key applications and recent developments related to Hamilton cycles in the field. Coming to the next slide, overview of Hamilton cycle. A Hamilton cycle is a closed loop that visits each node in a graph exactly once, without a repetition. Determining the existence of such cycle is an NP-complete problem, making it complete, computationally challenging. Despite this, and exact algorithms are used to identify Hamilton cycles in various bioinformatics applications. You can see you can see the example of Hamilton cycles G and G dash. These two are Hamilton cycles which repeats every vertex once. And coming to the next slide, biological sequence assemble. Hamilton cycles are used to model the sequence assembly problem, where DNA fragments are reconstructed into a complete genome. The problem involves finding a cycle that visits every fragment exactly once, representing the correct sequence order. This approach helps improve accuracy in assembling genomes from short sequence reads. You can see the some bio sequence, which is DNA sequence, amino acid sequence, and RNA sequence. DNA sequence promotes and enhancer DNA modification, and you can see in amino acid sequence, which is proteins and predicts, and DNA sequence, sgDNA and tGNA. And coming to our next slide, which is genome assembly algorithms. Deep Burgin graphs and overlap graphs are common structures where Hamilton cycles in are in genome assembly. Identifying Hamiltonian cycles in these graphs corresponding to reconstructing the original DNA sequence. Advances in algorithms have enhanced the efficiency to solve large-scale genome assembly problems. As you see the picture about the genome assembly algorithms. And moving forward to the next slide, which is protein structure prediction. As you see the protein structure prediction in the image, uh, and coming to its details, Hamiltonian cycle assists in modeling amino acid interaction within proteins, particularly in contact map analysis. Finding finding cycles helps identify stable protein conformation and folding pathways. This application contributes to understanding the 3D structure of proteins from their sequences. And moving forward to our next slide, this is about the protein structure prediction. And in our eighth slide, there is Phylogenetic tree reconstruction. You can see the image of the phylogical tree reconstruction, which looks like a tree, and Hamiltonian cycles helps in constructing phylogenetic networks by finding optimal paths that connect species based on genetic data. They assist in resolving complex environmental relationships where traditional trees are insufficient. Such cycles support the analysis of reticulate evolution and hybridization events. You can see the some image of the tree construction about phylogenetic, which consists of some parametric things. And moving forward to our next slide, which is metabolic network analysis. Metabolic pathway can be modeled as graphs where cycles represent continuous biochemical process. Detecting these cycles helps in understanding the flow of metabolics and enzyme interactions. These insights is useful for metabolic engineering and drugs target identification. You can see the picture of the metabolic network analysis as shown in the image, which is a network reconstruction in slice model of metabolism, and where it starts and how it works. This is the cycle of the metabolic network analysis. And moving forward to our 10th slide, which is a protein-protein interaction network. This is the protein-protein interaction network, how it's look like. You can see the phenotype A and phenotype B and nodes and how they interacted to each other. And Hamiltonian cycles are used to identify critical interactions pathways with complex protein networks. They help in pinpointing essential protein and interactions involved in cellular process. Such analysis can guide targeted either fits and diseases mechanism studies. Challenges in competing Hamiltonian cycles. The NP completeness of Hamiltonian cycle problem poses significant compu- computational challenges in bioinformatics. Large biological data sets require heuristic or approximation algorithms for practical solutions. Ongoing research aims to develop more efficient algorithms tailoring to biological network structure. And moving forward to our next slide, which is algorithmic approaches and heuristics. Then you can see the images, which is algorithmic SEO and heuristic SEO. Exact algorithms looks like black tracking and branch and bound array are used to small instance. Heuristic methods, including genetic algorithms and segmented analyzing, are applied to large and complex graphs. Hybrid approaches combine multiple stages to improve solutions quality and componential efficiency. This is about the algorithmic approaches and heuristics. Recent advance and tools. Recent advances and tools, which as you see some tools, visual impaired students, which is a voice input output system, accessible program environment, and screen manufacturing software. These are some recent advanced tools. And coming to its details, development of specialized software tools has facilitated the application of Hamilton cycle detection in biological data. Machine learning techniques are increasingly in, increase, integrated to predict the patience of Hamiltonian cycles. This advance enable more accurate and scalable analysis in bioinformatics research. And coming to our next topic, which is a case study in bioinformatics. Several case studies demonstrate the successful use of Hamiltonian cycles in genomic assembly and protein interaction analysis. For example, improved assembly of complex genomics has been achieved using Hamiltonian-based approaches. This example highlights the particle impact of graph theoretic methods in real-world biological problems. Coming to our next slide, which is future direction and challenges. You can see some challenges and future directions. Future research focuses on developing more scalable algorithms to handle growing biological data sets. Integrating Hamiltonian cycle methods with other computational techniques promises to enhance bioinformatics analysis. Addressing the computational complexity remains a key challenge for bo- broader applications. And coming to our last and final conclusion about this topic, Hamiltonian cycle provide valuable frameworks for solving complex biological problems in bioinformatics. Despite computational challenges, ongoing algorithmic innovations are expanding their applicability. Their continued development will likely contribute significance to advance in genomes, proteomics, and system biology. And thank you for this.